So please welcome Professor Adam Cohen.
Thank you. Thank you, Joshua, for the introduction, and I want to thank Michael for the invitation and all of you for being here today. So today I'm actually not going to talk that much about neuroscience or brain imaging. I decided that given the interests of the Allen Center, I would talk about pattern formation and chaos in bioelectrical systems. So before I do that, let's talk a little bit about what is bioelectricity. So this is us. If we zoom in by a factor of about 10,000, we see the cells that we're made of. If we zoom in by another factor of 100,000, we see that these cells have an inside and an outside, which are separated by a lipid bilayer, which is an electrical insulator, while the media on both sides are aqueous ionic conductors. Ions can move around. You can have an imbalance in the charge across this membrane, which will then lead to a voltage across this membrane. You can think of this a little bit like a parallel plate capacitor. So every cell and every organelle within every cell, every lipid-bound structure can support a voltage across its surface. This voltage can change as these ions flow back and forth across the membrane. And the electric field from this voltage pulls on every protein and every molecule associated with the membrane. So you can think of voltage like a global regulator of energy landscapes inside of cells and cellular organelles. A typical electric field in a neuron, say in our brain, is something like 30 megavolts per meter. Does anybody know what the electric field in a lightning bolt is? It's about 3 megavolts per meter. So the electric field in the membrane, in every neuron in your brain, is about 10 times bigger than the electric field in a lightning bolt. So if you feel like you're under stress, just keep that in mind. Okay? So these voltages are most famously studied in our brain and our heart, which are both electrically regulated organs. But in fact, membrane voltage is found throughout life. Here's a chart showing some of the voltages in different cellular structures. Bacteria have a voltage of about minus 100 millivolts. Mitochondria have a voltage of about minus 180 millivolts. This voltage provides part of the proton motive force, which drives protons through the ATP synthase, which is how we make ATP. So all of us are essentially electrically powered organisms. Some plants and fungi have gigantic membrane voltages, minus 250 to minus 300 millivolts, and nobody knows why that's the case. These have just been observed out in nature. Now, one of the challenges with studying voltage is that you can't see it directly. If you just look at a cell, you can't see the voltage in it any more than you could see the voltage in this wire by looking at the wire. And so for decades, people have been trying to develop some optical tools to convert the electrical activity in cells into an optical signal we can see. This could be useful for studying the heart and the brain, but it could also open up the scope of all of these other systems which have been very difficult to study with conventional techniques. Nobody has ever put an electrode into an intact bacterium before because bacteria are too small and they have a cell wall. Plants are also very hard to measure because they have a cell wall. Mitochondria are very difficult to access because the voltage is on the inner membrane there. If you have something with lots of cells and you want to map the voltage, that's hard to do with electrodes. Or if you have things that are moving around, like a sperm, it's hard to imagine chasing it with an electrode and harpooning it. Okay. So a few years ago, my lab became interested in this challenge of how to visualize electrical signaling in cells. The story starts here. This is a photo of the Dead Sea between Israel and Jordan. There's a single-celled microorganism that lives here that produces a protein which it uses to convert sunlight into a membrane voltage. It's a transmembrane protein that absorbs photons of sunlight and uses that energy to pump protons from inside the cell to outside the cell. That builds up a membrane voltage, which this creature then uses to power its metabolism, to power the ATP synthase. So you can think of this like the world's smallest photovoltaic device. Light comes in and a voltage comes out. So I said to myself, I wonder if we can run this protein in reverse. Instead of having light come in and a voltage come out, can we use the voltage in the cell to produce an optical signal that we can see? Okay? Now I'm going to skip 4 years of really hard work and tell you that we found a way to do this. We discovered that these proteins are a little bit fluorescent. It's a new class of fluorescent proteins with no relation to GFP or any other known fluorescent protein. They're excited by red light, and they emit in the near-infrared part of the spectrum. But they only emit when a proton is sitting on a particular functional group in the core of this protein. Now, the proton is charged, so if there's a change in the membrane voltage, that electric field can either push the proton on or pull it off of the protein, just like this. Okay? And you see this works in PowerPoint, so that's very encouraging. All right. Now I'll show you some data. Here's a human tumor cell where we're expressing this protein. And we're exciting it with red light and looking at the fluorescence. And I'll show you a movie where we apply steps in voltage between plus and minus 1/10 of a volt. What you should see is when the voltage goes up, the protein gets bright. When the voltage goes down, the protein gets dim. This response turns out to be very fast, less than a millisecond, and quite sensitive. By taking this protein and now expressing it in other cell types, we can start to make movies of electrical signaling in cells. Now here we have a passive reporter of the electrical activity in the cell. It's useful to be able to stimulate the cell with light as well so that we can have a bidirectional optical interface with our cells. To do that, we expressed a second protein, a channelrhodopsin protein, which acts as a light-gated ion channel. This protein is derived from a alga which lives in a freshwater pond in the south of England. So by shining blue light on the cells, we can activate this ion channel, which will trigger them to fire. And then by shining red light, we can elicit near-infrared fluorescence, which tells us about the firing of the cells. So this gives us a bidirectional interface to the cells. Here I'll show you a movie where we're looking at about 50 rat hippocampal neurons in a culture in parallel. And this movie is at 500 frames a second. And at a certain point, we'll turn on a blue light and stimulate all of the cells. The movie is slowed down about 10-fold from real time so we can see what's going on. And now you can see every action potential and every spike in every cell in the culture. The traditional way of measuring this electrical activity is to poke the cells with a little patch pipette, one cell at a time, which takes about 1 hour per cell. With these optical measures, we just recorded from 50 cells in 7/10 of a second. And so we can look at large numbers of cells and explore their functional behavior. One of the neat aspects of using light to interact with cells is that you can control when and where you shine the light. So rather than stimulating all the cells at once, we can go and target the light to stimulate just one cell or a combination of cells to see how these patterns then propagate through the culture. Here's an example of one of these cultures where we'll stimulate just this cell down here with a targeted flash of light. And then you see, as that cell gets stimulated, then there's excitatory and inhibitory behavior, which is elicited in other cells in the network. And so we can use this kind of technology to map the synaptic connections between different cells in the culture. And we're using this to explore synaptic biology and to screen for drugs that modulate synaptic function. We can also zoom in to the subcellular level and look in great detail at the signals within individual cells. We've developed some computational techniques to map the propagation of these electrical impulses with very high time resolution. Here's a picture of a single cell where we're stimulating it just here, and we're mapping the action potential wavefront, in this case, at 100,000 frames a second. And so here's what we see. Okay. And so you can really see in detail how these impulses propagate through individual cells. Now, I told you I wasn't going to talk about neuroscience, and I want to stick to that promise. So I'm going to tell you about some other systems where we see interesting bioelectrical behavior. We expressed one of these voltage indicators in E. coli, just regular lab strain of E. coli, and we looked at them in the microscope. We're not doing anything to them here. This movie is sped up about 6-fold from real time. We discovered that bacteria generate electrical spikes, a little bit like action potentials in neurons. This was a big surprise. As I said, nobody has ever put an electrode into a single intact bacterium before. We don't know how the bacteria generate these spikes or why they do it, but this seems to be a common phenomenon in all of the bacteria that we've looked at. And so there's something interesting going on with bacterial metabolism and electrical signaling. And the field of bacterial electrophysiology is entirely unexplored. There are millions of species of bacteria in the world, and nobody knows anything about the electrophysiology of any of them. We've also looked in zebrafish. Here's a 3D reconstruction of a zebrafish heart, looking at the electrical wave propagating through the heart inside of the live animal. We've been doing this to study the embryonic development of the zebrafish heart, to see how does this organ boot up. at the beginning, we have a single cell which has no heart, and then about a day later, this animal has a heart which is beating, and we want to see how it comes into being. And finally, we're doing a lot of work in mice. Here's an example looking in the brain of a mouse. These are recordings from 2 neurons in this brain, and as the mouse starts walking, we can see it, these neurons start to spike away. Okay, so in the remaining time, I want to tell you about a sort of offbeat application of these tools towards trying to create synthetic bioelectrical systems where we start with cells that are electrically inert, and we introduce components one at a time to try to imbue these cells with interesting bioelectrical behavior. So as opposed to disassembling complex naturally evolved systems, we're going to take a synthetic approach and try to create things from scratch. So in the famous Hodgkin-Huxley model of a neuron, you have a sodium channel and a potassium channel, and those are enough to cause the neuron to spike. So we took a line of human tumor cells that started electrically inert, and we introduced 4 transgenic components into these cells. One is a sodium channel. The second is a potassium channel. The third is this light-gated ion channel, which we can use to trigger the cells. And the fourth is this voltage indicator so we can see what's going on. But there's only 2 active components here, the sodium channel and the potassium channel. We then stimulated these cells with flashes of blue light of successively greater intensity. And for the dim flashes, we saw corresponding small changes in voltage. But then once the flash crossed a threshold, we saw these big spikes in voltage, which are action potentials. And so, indeed, as everybody expected, with just these 2 components, you can create an electrical spike in a cell that didn't have that property before. But unlike in a neuron or a cardiac cell, we really know everything there is to know about these channels. And so we really understand how these cells are making these spikes. Or at least we thought we did. So one day we were looking at a culture of these cells, and we stuck a patch pipette into one of the cells and recorded the voltage. And this is what we recorded. So the cell was spiking away. Now, I explained how this sodium channel and this potassium channel can drive a spike. But it was a mystery to us why the cells kept spiking over and over again. What's re-triggering them after each spike? Why is this behavior periodic? We then put a voltage indicator into the cells, and we looked in the dish. I'll show you a movie of what we saw now. This is about 6 millimeters on a side, so there's about 100,000 cells here. You can't see the individual cells. And this is a map of the voltage in this dish as it's sitting there. So looking at a single point with this patch pipette, we saw this periodic behavior, which really corresponds to this spiral wave sweeping over the cells over and over again. So what's going on? These cells couple one to their neighbors through endogenous gap junction proteins, connexins, which create an electrical coupling between the cells. So when one cell fires, it can trigger its neighbors, which can trigger its neighbors, and so on. And this leads to these waves, and then the waves can sweep around and periodically re-trigger the cells. So this is an emergent phenomenon which only happens in the large population of cells. Isolated cells don't spike like this. So then we started to explore these electrical waves. We developed techniques to pattern the growth of the cells. So here we have a little snake of the cells, and we'll stimulate them up here near the top. And so you can produce these waves of cells, which can electrical waves, which can propagate through these spikes. Through these snakes. You can also pattern the propagation of these waves just using light. So here's a contiguous layer of cells, and then we'll stimulate it just with a little flash of light in the center. And we can induce these outward going waves of electrical activity. If you pattern the cells into a annulus, into a ring, we've developed techniques for launching directional waves in these rings. So here's the initial wave, and then we'll launch it. And now these waves will go around and around in these rings, and they'll chase their tails. They'll do this for hours and hours, tens of thousands of revolutions going around here. Okay, so now why is this useful? Well, by having very long measurement times of these waves propagating in the dish, we can measure the frequency extremely precisely, and we can use this to detect very subtle perturbations in the physiology of the cells. I call this, half-jokingly, our cellular cyclotron, right? So by looking at these things going around and around, we can measure their conduction attributes very precisely. We can also optically define these cellular ring oscillators. Here I'll show you an example where we use steady blue illumination to inactivate the sodium channels in this track-like pattern so that we have just a conductive track with inexcitable boundaries on the outside and the inside. And then we'll launch a wave which will go around and around in the track. That's setting up the track, and then there's an initiation pulse, which will come in a second, and then we'll launch this directional wave. And now this wave is going around in the optically defined boundaries. One of the neat things about having optical boundaries is that you can reshape them. And so now we're going to squish these boundaries down and make this wave go around faster and faster. Okay? We've been interested in using this to try to explore what are the conditions that lead to arrhythmic or irregular behavior. So in the heart, of course, this is a major medical concern. When people have arrhythmic behavior in the heart, it can be fatal. And it's challenging to understand in the heart because there are so many different ion channel components which contribute to the behavior. We thought, well, at least in this simple system where there's only two components, we ought to be able to understand when do we get arrhythmia and when do we get regular behavior. It turns out, even in this very simple system, when you have just two components, but you have thousands of coupled cells, the dynamics can be surprisingly complex, and the transitions to chaos can be actually quite difficult to understand. Let me give you an example. Here we're looking at a serpentine pattern of cells. So they're connected off the field of view here, as well as we have several islands of cells here. We're going to now stimulate these cells periodically and launch waves going down these cells. And so you can see these islands down here are blinking away, and we have these waves going back and forth along the cells. And there's a little break here, so the pattern ends. And now we're going to do this at different frequencies, and we're going to look at the patterns that emerge in these cells. Here are the 0D islands, where we stimulate them at different frequencies. As we go to higher and higher frequencies, we see that these waves just gradually disappear, and eventually the cells cannot keep up with the high-frequency waves. Interestingly, in the region where we stimulate the cells, as we go to higher frequency, we get what's called an alternans behavior, so an alternating big beat and a little beat. In the human heart, this is often a precursor to arrhythmia. Cardiologists will diagnose this, and if they see this, it's a warning sign. And then as we go to higher frequencies, we get this chaotic dynamics here. If we look in the far field, so as these waves conduct, this alternans pattern disappears. And at the highest frequencies, only 1 out of every approximately 5 waves actually makes it into the far field, and you come back to a regular pattern. And so this very, very simple system is letting us now explore what are the parameters which lead to either irregular or regular beating in these cellular oscillators. And they're suggesting that there are interesting geometrical effects which are important in the regularity, totally independent of the things that we usually think about, which are the patterns of gene expression and the details of the ion channels. All of the cells here have exactly the same ion channels in them, and the only difference between these different situations is the geometry and the boundary conditions, which then govern what kind of dynamics you get. Great. So when Michael invited me to come talk, he said that I should try to be provocative. And so I want to end with some wild speculations, which are not substantiated by any evidence, which I hope will provoke some thought among all of us. I want to talk about a very surprising historical and mathematical coincidence, which I recently came across. So in 1952, Alan Turing published this very famous paper on the chemical basis of morphogenesis. He showed that if you have chemicals which can diffuse and can react with each other, under certain mathematically well-defined conditions, you can get stable patterns of chemistry which are reminiscent of the kinds of patterns that you find in life. So stripes and dots and lots of other patterns that you see for instance, in these big cats, or you see them in fish or in seashells. One of the requirements for this kind of dynamics to emerge is that you have to have at least two things diffusing and reacting. And then there's other constraints on the diffusion coefficients and the details of the reaction. But there's a precise statement of what's needed. Now, remarkably, at the very same time, Hodgkin and Huxley published their mathematical model of the nerve. Just look at the dates here. This was received 10 March 1952. This was submitted the second time, 15 March 1952. If you look at the Hodgkin-Huxley equations, they actually have exactly the same mathematical structure as the reaction diffusion equation from this Turing model. So here you have a first derivative with respect to time is equal to a second derivative with respect to space plus some nonlinear term. Now, if you have only voltage, we know that you cannot get stable patterns like this. But if you have two quantities, say voltage and calcium, then at least mathematically, the analysis here is the same as the analysis here, and there will be some conditions on the ion channels which will give you stable patterns of voltage where you have regions with high voltage and regions with low voltage which are static there. Okay? And so the question is, this is mathematically possible, does it happen? So if you look at the Turing model of morphogenesis, depending upon the values of the parameters in those equations, you can get different kinds of dynamics. And you can classify them as 6 different types. There's the most boring, which is nothing's happening. You can get uniform oscillations. You can get stationary waves with very high frequency, so where like from one cell to the next things oscillate. You can get oscillating waves with very high frequency. You can get propagating waves, and you can get these stable patterns. Now, if we look at electrophysiological systems, we see 3 of these that are known. Of course, there's plenty of boring stuff in the world where nothing's happening. We've seen oscillations where cells can vary their voltage spontaneously without any spatial component. And I just showed you a whole bunch of examples of these excitable media where we get propagating waves. This is a solution to the Turing model, but this looks just like those spiral waves that I was showing you in the electrical system. I don't know, and I don't think that anybody knows, whether it's possible electrically to get class 6 over here, which are these patterns, which don't change in time, but have very interesting spatial structure. So I'm going to leave you with a homework exercise, which will be due at the next meeting of this Discovery Center, which is to identify a combination of ion channels whose conductances meet the conditions that Turing wrote down in 1952 for forming one of these stable patterns. And first to show by numerical simulation that these channels can lead to these patterns. The second part of the exercise is to express these channels in a cell line of your choosing and to show experimentally with some voltage imaging that these Turing patterns actually emerge. And then the third part for extra credit is to identify a situation where this actually happens in a living pattern-forming system. Okay, so those will be due at the beginning of the next meeting. So finally, I want to thank all the people who've done the work. I've had a fantastic group of students that I'm working with. Almost all the work that I showed today on these synthetic systems is the work of Harry McNamara, who's a graduate student in my lab. These are some of our collaborators. These people pay the bills. This is my lab, and I'm happy to take questions. Thank you.
So is an electroencephalogram, is an electroencephalogram one of your Turing patterns?
One of our what?
The EEG.
Is it a Turing pattern? Yeah. Well, so in the brain, the dynamics are much more complicated than what I wrote down here. And so the EEG is a aggregate measure of the roar of the crowd of all of the neurons in the brain. And of course, there are very complex spatial patterns that arise in natural neural activity. But I think that is a more complicated situation than what I've been discussing. In the case that I was talking about, the cells just have simple nearest neighbor couplings, and that's it. And so we don't have we don't have neurotransmitters or any of the more complex synaptic dynamics that you would find in the brain. Yeah.
Have you tried altering the stoichiometric ratio of the two channels that you use to see if that affects pattern income?
Yes. So it turns out there are many interesting things that happen as you change the conductances of these different channels. We've been using that effect to screen for drugs that modulate these channels. So for instance, there's a sodium channel, NAV 1.7, which is very important in mitigating pain sensation. And using these spiking cells, we can look for compounds that modulate the excitability of the cells, and that by blocking this channel. And that actually works. We've been screening large libraries now to look for new modulators. In addition, there are interesting qualitatively different things that will happen as you add drugs. So sometimes drugs can cause transitions from regular spiking to chaos. There can also be other kinds of phase transitions or dynamical transitions in these cells, which can be driven by specific drugs, which can, for instance, flip them from one stable voltage to a different stable voltage. So there's a lot of interesting things that happen even with these very simple systems. Yeah.
How is the chaos is damping when you go to start on the, how do you dampen?
How is the chaos damping? Yeah. So if you look at those waves as they go through those 1D tracks, what's happening is that the little spikes that are riding on the tail of a bigger spike, the sodium channels are inactivated on that tail, and so those little spikes get suppressed, and then the bigger spikes win. And so there's an instability, which is a winner-takes-all instability. And so one wave will win, all the others will get suppressed, and that will be the one that propagates. And then the next, after that one has propagated, the next one that comes along will see fresh cells and will propagate again, and then the ones after that become inhibited. Yeah, in the back.
So you mentioned these microorganisms and yeah, and those channels and the dead sea microorganisms, and you compare them to sort of photovoltaics.
Yeah.
Is anybody using or developing these as photovoltaics or as any kind of energy device?
It's an interesting question. So people have contemplated this. It turns out that they're quite inefficient, and so as a practical matter, it probably wouldn't be competitive with the other kinds of things that are out there. You can certainly get a voltage out of them, but it's very, very small. In the 90s, there was a lot of interest in using proteins of this family for different optoelectronic applications. They have very interesting optical nonlinearities, and so people thought about using them for data storage and other kinds of devices. None of that ever panned out in a practical matter, but their application in neuroscience and biotechnology turns out to be quite interesting.